So the ALLG is Australia's and New Zealand's only not-for-profit blood cancer clinical trial research group, and we've been running initially as the lymphoma group and the leukemia group as two separate groups for 45 years now, and conducted 150 clinical trials with more than 6,000 patients. And I hear the little sniggers from the intensivists in the room. Our cancers are rare cancers. Our trials are small patient populations. In fact, for many trials, we'll have as many investigators as we have enrolled patients amongst our 750 clinician, scientist, nurse, and data manager members. There are 7 disease groups, which I'll tell you about in a little while. And as of about 5 years ago, we separated our scientific advisory committee from the board who provide our overall governance for the ALLG. And it's the scientific advisory committee who just do that, who are responsible now purely for the scientific direction of the group. The purpose, the vision of the ALLG is better treatments, better lives, and in particular through delivering clinical trials to improve the outcomes of patients with blood cancer. The ALLG Trials Centre runs out of Melbourne with 19 staff, and all our trials and registries, the National Blood Cancer Registry, now have an electronic data platform, which was itself a 2-year mission. We commence about 3 or 4 new trials per annum, and we usually have about 16 trials open to recruitment. And because there is a very broad spectrum of very rare blood cancers, I would suggest that diffuse large B-cell lymphoma is our only common cancer and all the rest are rare. We organise our work around these common blood cancer classifications and have different sort of craft groups, the acute leukemia and the chronic leukemias, the aggressive and the low-grade lymphomas, myeloma, and then our transplant and supportive care studies. And we all feel very much excited and very much a part of what has been a great privilege as a haematologist in the last 10, 15 years to be a part of an extraordinary revolution in targeted therapy as we move more and more away from the blunderbuss chemotherapies that we used to treat everyone with. We've had highlights listed there, 15 conference presentations in the last year and 8 journal publications, and 3 recent successful MRFF grants. We currently have 16 recruiting trials, and our 2 scientific meetings that we hold per year are very highly ranked in terms of education, because not only do we present what is in the proposed and the current trial portfolio and what's been recently published, but we also provide sort of up-to-date educational sessions in each craft group. And then there are 11 contributions made to government in terms of consultations. I must say myself, I feel a bit ambivalent about these advocacy. We have to be very, very careful that often pharmaceutical industries are very keen for us to advocate on their behalf. And I see that the Pharmaceutical Benefits Advisory Committee do a very good job without us necessarily jumping in and helping them assess the data. And obviously low survival rate cancers has been a big focus of ours at the moment. So I've been a little partisan here because I'm predominantly a lymphoma clinician researcher. So I'm going to show you some examples from our international lymphoma collaborations. We joined with the UK, with Cancer Research UK funded study, the RATHL, Response Adapted Therapy in Hodgkin Lymphoma trial, using interim PET to adapt therapy for patients, trying to maximise their chances of cure and minimise their chances of toxicity, in particular infertility in these patients who are quite young for the vast majority of patients with Hodgkin lymphoma. And now, you know, with a 5-year survival approaching 94% in Hodgkin lymphoma, RATHL has certainly now become a standard of care for patients, particularly those who want to preserve their fertility. In my own personal experience, having been a participant in the PRIMA study of rituximab maintenance after first-line treatment for follicular lymphoma, I did an exploratory analysis of PET scanning in the context of that study, hoping there would be enough renegade physicians like myself who had conducted PET scans in these patients. That was the first of what was subsequently a large collaboration and a pooled analysis of 3 international trials looking at the prognostic value of PET after first-line treatment for follicular lymphoma, which then, on the basis of that data, had the impact of encouraging Roche to spend probably over $100 million incorporating PET scanning back in 2011 in their large 1,400 patient follicular lymphoma gallium study. And this is PET analysis in that study has really finally secured the role of PET as the gold standard for response assessment over CT scanning in patients after their first-line treatment of follicular lymphoma. And it was that data that assisted me in that sort of sequential building of the data and pooled analyses, and then the subsequent prospective large study, which helped me with a MSAC application. And anyone who'd like to hear what it's like to be a sole clinician applicant for MSAC, I had what was called a fit-for-purpose pathway, and God forbid anyone who doesn't do a fit-for-purpose pathway, because it was a very prolonged but very enlightening 2-year journey in terms of getting MBS listing of PET for indolent lymphoma here in Australia. And this has established PET as the platform for response-adapted therapies, trying to improve the outcome for the 20% of patients who have a very poor prognosis if they remain PET positive after their treatment. And it's enabled us to now join in with the Portray study to try and do this. Looking beyond my own interest in acute myeloid leukemia, that's certainly been one of the flagship diseases for the Australasian Leukemia Lymphoma Group, and several trials over 3 decades have really contributed to the development of AML protocols, the recipe for treating acute myeloid leukemia globally. And our largest randomized trial was of 444 odd patients, which unfortunately, while it showed a significant increase in the leukemia-free survival with increased cytotoxic, increased anthracycline in consolidation for these patients, because of the morbidity and mortality of this, there was no actual impact on overall survival. Acute promyelocytic leukemia, this was a study that was published in, I think, 2012, the APML4 trial led by Harry Ireland at the University of Sydney and Royal Prince Alfred Hospital, where we were combining all transretinoic acid with arsenic and chemotherapy. And that was one of the precursor studies to a chemo-free regimen using ATRA and arsenic alone to cure acute promyelocytic leukemia. And I think it would have been quite special to have had that cartoon on the front of Blood along with the editorial about the APML4 study. And this most rapidly fatal acute leukemia has now become more than 90% curable with chemo-free approach. And so now in APML5, we're actually looking at trying to see if we can deliver oral rather than intravenous arsenic and greatly reduce the impact of daily intravenous arsenic infusions in an ambulatory care setting for patients with APML. I think many of us in the cancer sphere will remember the Time article with the magic bullets for cancer some years ago. The contribution that members of the Australasian Leukemia Lymphoma Group, and in particular Tim Hughes and all the younger clinician researchers that he and Sue Bramford in the laboratory at Royal Adelaide Hospital have achieved is really at the forefront of the management of CML globally. Previously incurable, it's probably the first cancer that's curable through the use of a single targeted agent with the tyrosine kinase inhibitor imatinib, and now through successive trials we've been able to show that not only can we achieve deeper molecular responses with the newer generation tyrosine kinase inhibitors, we can actually now successfully stop treatment, which will be of great joy to PBAC, when you think of the successive numbers of patients year in, year out who are on these tyrosine kinase inhibitors. But I also want to acknowledge that it was the work with the collaboration of all the centres in the ALLG providing specimens to the Royal Adelaide Hospital that really enabled Sue Branford's lab to develop accurate, sensitive molecular response assessment, and it's actually placed Royal Adelaide as one of 3 reference labs internationally for studies in CML. And it's really quite remarkable that in my career we've gone from a survival curve that was really almost 0 at 5 years to up there over 9% at 5 years. But there have been challenges, and there continue to be challenges. The very rapid pace of developments in haematology, combined with slow recruitment given our population base within Australia and New Zealand, has limited the clinical applicability of some ALLG trials. And all of us successful investigators can certainly add to our CV of failures with trials that haven't recruited well. Andrew Spencer leads a large portfolio of myeloma trials, and the challenges with thalidomide maintenance only being available and no lenalidomide maintenance available creates challenges for the design of trials in myeloma. Interim PET adapted therapy, NHL 21, the international PET criteria for response assessment changed just after this trial completed recruitment, and low-dose chemotherapy for the elderly in chronic lymphocytic leukemia has been surpassed globally by extremely well-tolerated oral enzyme inhibitors. So the challenges and impact very much relate to trial funding and recruitment because almost all of our cancers are rare cancers. But there is no doubt that the MRFF rare cancer funding will hopefully be a big boost to the ALLG's trial portfolio, with 3 successes in 2007 and 4 submitted in 2018. The key gaps are the support that we need for our clinical trials so that we can ask the questions that are of relevance to our patients and our investigators, not the questions that are relevant to the drug companies who are often supplying us with the agents for these trials. But also we do have to have a mature and very robust partnership with pharmaceutical companies in drug development. I'm going to show here, and this is not an ALLG initiative, but it really did spawn from the Haematology Clinical Research Network, the ALLG data managers from New South Wales. And you can see up there, ClinTrial Refer Haematology New South Wales was the very first ClinTrial Refer app that has rapidly been replicated, as you can see there, with more coming. And this is the simple app that helps the clinician and indeed the consumer identify within a minute what trials are currently open and recruiting and what their inclusion criteria are. And that has been a very useful tool for us in haematology, which increased in New South Wales 8-fold the cross-referral of patients to various sites, because patients really don't care about what local health district you work for. They're quite happy to go to whichever hospital will provide them access to the cutting-edge trial, and has resulted in a 30% sustained increase in recruitment over the last 5 years. The other challenge we have in documenting implementation of both ALLG and global trial data is the increasingly variable application of practice of multidisciplinary team meetings in Australia, and that's obviously very, very fashionable and very important for a good reason. It's very fashionable in haematology and oncology, but we have variable SOPs and documentation of this, and I think there's enormous potential perhaps within the ALLG to be collaborating and maintaining currency of pro formas across the different OMIS systems. So the ALLG, in its commitment to better treatments and better lives, I think it's fair to say we've been continuously focused on the impact of our trials, but we certainly need to start getting more serious about the potential to focus on implementation. Thank you very much.